Hello friends, this is the second video of the glucose metabolism, and in this video we will discuss this glycolysis. So what is glycolysis? Glycolysis is a pathway where 6-carbon glucose is split into two molecules of pyruvic acid under the aerobic condition or the lactic acid under the anaerobic condition. It is also known as the Embeden-Meyerhof-Barney because it was discovered by Embeden and Meyerhof. So next we will discuss about the significance of the pathway. What are the significance of the glycolysis? Glycolysis is the only pathway which take place in all the cells of the body or all the organism, whether it is eukaryotic or whether it is prokaryotic. It is the only source of energy in the RBC because RBC lack the mitochondria. So in RBC, anaerobic respiration takes place and glycolysis is the only source of energy for the RBC. And third, it is the major source of energy for the muscle during the strenuous exercise. When we exercise, then oxygen supply is not adequate. During that time, formation of lactic acid occur and the production of ATP via the glycolysis occur, and it is the main source of energy during the exercise, and it is the preliminary step before the complete oxidation of glucose. We know that during the complete oxidation of glucose, it required for we require acetyl coenzyme A, which is formed from the pyruvic acid, and pyruvic acid is from the glycolysis. So it is the preliminary step before the complete oxidation of glucose metabolism. Step first of all, what is the step take place during the glycolysis is the entry of the glucose. As we have discussed yesterday, there are various glucose receptor. So GLUT4 and GLUT2 are present majorly in the cell, and GLUT4 is insulin dependent, means when insulin is present in the cell, it will allow the entry of the glucose, and GLUT2 is not insulin dependent. Okay, next we will discuss the cycle. So glucose is converted to glucose 6 phosphate by hexokinase or glucokinase. These are two different enzymes. Further we will see the difference between these two enzymes. Here the utilization of 1 ATP takes place and formation of glucose 6 phosphate occur. Then by the isomerase enzyme, glucose is converted to fructose, so glucose 6 phosphate is converted to fructose 6 phosphate. Then in the third step, fructose 6 phosphate is converted into fructose 1,6 bisphosphate by phosphofructokinase. Here also utilization of the energy occurs. This is the key enzyme or the rate limiting enzyme of the glycolysis. Then from the fructose 1,6 bisphosphate, aldolase A form the glyceraldehyde 3 phosphate, which is isomeride to the dihydroxyacetone phosphate. And this glyceraldehyde 3 phosphate, in the presence of glyceraldehyde 3 phosphate dehydrogenase, with the reduction of the NAD plus to NADH, form the 1,3 bisphosphoglycerate, and 1,3 bisphosphoglycerate by bisphosphoglycerate kinase is converted to 3 phosphoglycerate by the utilization of by the formation of 1 ATP, and 3 phosphoglycerate is converted to 2 phosphoglycerate by phosphoglucomutase. So what takes place in the conversion of what is happening in the 3 to 2 conversion, thus the position of the group is changing. So always remember that when the position of the group is changing, always mutase will come into effect. So 3 phosphoglycerate is converted to 2 phosphoglycerate by phosphoglyceromutase, and 2 phosphoglycerate by the enolase enzyme is converted to phosphonyl pyruvate. And this is the step where the fluoride act, and fluoride inhibit the utilization of glucose and formation of phosphonyl pyruvate occur. Then phosphonyl pyruvate by pyruvate kinase where the kinase occur, there is the utilization or formation of ATP. So here the formation of ATP occur and the formation of final product of glycolysis, that is the pyruvate. So let us revise the cycle. That glucose is formed: glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bisphosphate, then glyceraldehyde 3 phosphate, 1,3 phosphoglycerate, 3 phosphoglycerate, 2 phosphoglycerate, and phosphonyl pyruvate and pyruvate. Uh, as this pathway is consists of the 10 step, it is difficult to remember in once. So best way to remember that this cycle is try to write and then remember. If you will learn and write and learn 2 to 3 times, you will easily remember this cycle. Next we will discuss about the some of the important points of the glycolysis. First of all, irreversible steps of glycolysis. So the step which is catalyzed by the hexokinase, phosphofructokinase and pyruvate kinase is irreversible. Let us see the steps. So first step which is catalyzed, that is conversion of glucose to glucose 6 phosphate is irreversible. Then fructose 6 phosphate to fructose 1,6 bis phosphate is irreversible, and phosphonyl pyruvate to pyruvate is irreversible. So these three steps are irreversible in the glycolysis. Step number 3, that is, I have already told you that the step which is catalyzed by phosphofructokinase is the committed step, rate limiting step, and it is a key enzyme of glycolysis or the bottleneck on enzyme of the glycolysis. And third important point is that fluoride inhibit the enolase activity. I have already told that second last step which is catalyzed by the by enolase, it is under the inhibition of the fluoride, and it is used in the blood collection during the glucose metabolic estimation. That if we take out our blood and for the glucose estimation, and our RBC will metabolize the glucose, then the glucose level will fall and the test will be altered. So in this condition we use the fluoride which will inhibit the enolase, so the glycolysis is inhibited and glucose will not be utilized and our test will be perfect. Third point that we have to discuss is about the energetics. We have seen that the two molecules of ATPs are utilized in the above step and four molecules of ATP are produced. So net two molecules of ATP are produced, and two molecules of ATP two molecules of NADH is also produced, which will give rise to the 5 ATP. As we know that one NADH to give rise to 2.5 ATP, so 2 NADH give rise to the 5 ATP, and this will occur during the aerobic respiration when the electron transport chain will function. So total, so you have to carefully read and answer whether it is asking for the aerobic or anaerobic. So total ATP production in aerobic is 7 ATP, that is in the presence of the oxygen, 5 ATP from the NADH plus, and 2 ATP from the ATP, and under the anaerobic condition only the 2 ATP form. So uh, under aerobic condition NADH go into the electron transport chain and will form the 5 more ATP. Next we will discuss the regulation of the cycle. So regulation of the cycle is first of all by the regulatory enzyme, that is glucokinase, phosphofructokinase and pyruvate kinase. So first is the glucokinase or hexokinase. These are under the influence of the insulin, and I have told you that I will told you the difference between the glucokinase and hexokinase. So this may come in the exam that hexokinase is present in the all the tissues. Hexokinase is present in all tissues, while glucokinase is only present in liver. And affinity to the, we measure by km. Hexokinase has the very low km. Very low km means it has very high affinity. So hexokinase can work in very low concentration of glucose, and glucokinase have very high km, so it cannot function in very low concentration of glucose, means it has a low affinity for the glucose. And hexokinases act on the glucose, fructose and mannose, while the glucokinase act only on the glucose. Induction, next we will talk about the induction by the insulin. So hexokinase is not under the influence of the insulin, but glucokinase is under the influence of the insulin. Now, what is the function of hexokinase and glucokinase? Both are the glycolytic enzymes which increases the glucose metabolism or the glycolysis. Next, we will discuss the phosphofructokinase. Phospho, just this all is about logical. Thus, ATP and citrate are the most important inhibitor. Now, why the ATP and citrate are the inhibitor? As ATP, if there is more ATP in the cells, then we do not need more formation of ATP. and we need to inhibit the ATP formation. So ATP alloristically go and binds to the phosphofructokinase, so it will inhibit. Okay, and citrate is an intermediate of the citric acid cycle. So that if there is more citric acid cycle going on and citrate is forming and not being enough utilized, then citrate goes and inhibit the phosphofructokinase. So these two are the modifiers or the inhibitors of the phosphofructokinase. And AMP, when ATP is utilized, AMP is formed, and AMP act as alloristic activator, because when AMP is there, then ATP level falls in the cell. So AMP goes and bind to phosphofructokinase and it activates the enzyme. Next, fructose 2,6 bisphosphate also increases the activity of phosphofructokinase. Now I'll here is the formation of fructose 1,6 bisphosphate. Now you'll think that why, where is the fructose 2,6 bisphosphate activating? This is the enzyme phosphofructokinase 1. There is one more enzyme phosphofructokinase 2 which will form phosphofructokinase 2, which will form fructose 2,6 bisphosphate, and this fructose 2,6 bisphosphate is the activator of phosphofructokinase. So indirectly we can also say that phosphofructokinase 2 is indirectly activator of the phosphofructokinase 1. So this, by this way, phosphofructokinase 2 and fructose 2,6 bisphosphate is also the activator of phosphofructokinase. Next enzyme is pyruvate kinase. Pyruvate kinase catalyzes the irreversible step of the glycolysis, that is the last step of the glycolysis, and insulin increases the activity of pyruvate kinase and glucagon decreases the activity of pyruvate kinase. Pyruvate kinase is inactive in phosphorylated state. Here we can see this most of the enzyme of the glycolysis is inactive in the phosphorylated stage. How? When there is entry of the glucose into the cell, then glucose means it increases the formation of the ATP. So the ATP phosphorylates the enzyme. When there are high levels of ATP, then automatically the enzymes of the glycolysis uh, lower their effect and there is low utilization of glucose. And when there is more AMP, means less ATP in the cell, then these enzymes can not be phosphorylated, so it activates the enzymes and it form the more ATPs. Third, it is under the hormonal control. Fourth is under the hormonal control of insulin and glucagon and glucocorticoids. Glucocorticoids are the hormone which increases the blood glucose level. And so insulin activates the glycolytic enzyme and increases the utilization of the glucose. And secondly is glucagon and glucocorticoid. They inhibit the glycolysis and they increase the gluconeogenesis, that is they wants to increase the blood glucose level. A normal blood glucose level is 70 to 100, in the fasting condition is 70 to 100 mg per dl. So when blood glucose fall, the glucagon and glucocorticoids and other hormones, they increases the blood glucose level by inhibiting the glycolysis and utilization of the glucose by these cells. Okay, so in next video we will learn about the next cycle, that is Cori cycle, and the Rapaport-Luprin cycle, and the pyruvate dehydrogenase complex. This in this video we have only seen glycolysis, which can come as a long answer, and you can extend these points to fill your more details, and also refer to the textbook for the proper usage. Thank you.